Hello students. Today I will be giving a brief presentation on Introduction to Gastrointestinal Tract and Physiology of Salivary Glands. So let us quickly see what all the topics we are going to cover in this session. The first topic will be basic physiological anatomy of the gastrointestinal system, like how the GIT is innervated, what is the nerve supply of the gastrointestinal tract, and basically we will be discussing about the enteric nervous system here. Then there will be discussion about the general principles of the gastrointestinal functions, and later on in brief we will be discussing about the physiology of salivary glands, that will include secretion and composition of saliva, control of the salivary secretions, and functions of the saliva. So let us start. Gastrointestinal system, also known as the digestive system, is like a portal or a door via which various nutrients, vitamins and minerals as well as fluids enter the body. So the gastrointestinal system acts like a doorway to the inner human body. Now, digestion is defined as the process by which food is broken down into simple chemical substances that can be absorbed and used as nutrients by the body. So digestion means basically breaking down of complex food into simpler compounds. Gastrointestinal system comprises of majorly alimentary canal that starts from the esophagus and ends at the anus, and other associated organs such as the salivary glands, the tongue, the liver, the gallbladder and the pancreas. The alimentary canal is a hollow muscular tube measuring around 10 meters or 30 feet that extends from the mouth to the anus. It comprises of mouth, tongue, teeth, pharynx, esophagus, which is 25 centimeter in length, the stomach, the small intestine, which have three parts again, duodenum, jejunum and the ileum, the large intestine consisting of the ascending colon, the transverse colon and the descending colon, and the lowermost part, that is the rectum and the anus. Here in this pictorial representation you can see the parts and the structures of the alimentary canal. So it starts in the oral cavity, which consists of the tongue and the teeth, which helps in chewing the food and mixing the food with saliva so that the bolus of food can easily pass through the esophagus. Now down the esophagus the food will enter into the stomach. This is the fundus of the stomach, this is the body, and this is the pyloric antrum. Now after passing through the stomach, the food or the bolus will enter into the intestine, that is the small intestine, and here it will pass along via the ileum and enter the ileum, and it will enter the colon via the ileocecal valve. Then the chyme will move through the ascending colon, the transverse colon and the descending colon, and ultimately it will be stored in the rectum and it will be eliminated from the body whenever required via the anal canal or the anus. The functions of the gastrointestinal system can be broadly divided into digestive functions and non-digestive functions. So let us see the digestive functions of the GI system. First is ingestion of food. So GI system involves placing the food in the mouth, that is eating the food, then chewing the food with the process known as mastication, lubrication and moistening of the food, and swallowing. Digestion of the food is the second function of GI system that converts complex insoluble large organic molecules into soluble smaller and simpler form, which can be easily absorbed by the body. The third function is absorption of those digested food. Now movement of these digested molecules from the lumen of the alimentary canal across its epithelial lining towards the blood or the lymph is known as digestion. So when the food is broken down into smaller components, the food is mixed with certain digestive enzymes, then the food moves down the alimentary canal and the nutrients are absorbed as the food moves through the alimentary canal, and ultimately these nutrients reach to the blood or the lymph. The fourth function is egestion or excretion of the unwanted products. So, excretion of the undigested food by the alimentary canal is done in the form of defecation. The non-digestive functions mainly include the immune system. There are lymphoid tissues present along the alimentary canal in the form of tonsils, adenoids and Pierce patches. So, especially the Pierce patches, which can be seen in the small intestine, they constitute an important part of the body's immune system. These patches, Pierce patches, tonsils, this group of lymphoid tissues, basically they provide both kind of immunity, that is humoral as well as cellular immunity. So it is very effective against the microbes which are trying to enter our body via the alimentary canal. Now let us see the structure of the gastrointestinal wall. In general, the wall of the gastrointestinal tract is formed by 4 layers. Basically, the alimentary canal, the entire alimentary canal which starts from the esophagus till the anus consists of these basic 4 layers from inside to outside. So, as we see from the inside of the gut towards the outside, we will find these 4 layers in the gastrointestinal wall. So first layer is the mucosal layer or the mucus layer. This mucus layer will again have 3 different layers coming from inside to outside the gut, that is the surface epithelium. Then the second layer will be the lamina propria. The third layer is muscularis mucosa. Now, after the mucosa, when you go towards the periphery, towards outside of the gut, you will find submucous layer or the submucosa. The submucosa is very important because it contains a special nerve plexus known as the submucosal nerve plexus or the Meissner's nerve plexus. Now, after this layer, you will find another layer known as the muscular layer or the muscle coat. Now this, as the name suggests, this layer will consist of 2 different kind of muscles. So the entire GI wall is made up of 2 different types of muscles. The inner layer of the muscular coat will contain the circular muscle fiber, the circular smooth muscle fibers, and the outer layer of the muscle coat will contain longitudinal smooth muscle fibers, and in between these inner and outer smooth muscle layers there is another type of nerve plexus which is very important for the GI motility is present, known as the Orbach's plexus or the Meindrick plexus, and the last layer or the outermost layer of the GI wall is known as the serosa or the fibrous layer. Here you can see a cross section of the alimentary canal which depicts the various structures of the gastrointestinal wall. So the innermost layer, as I have mentioned, the innermost layer is the serosal layer or the serosa. So the serosa will contain the first layer, that is the epithelial lining, the epithelial layer. This yellow color structure which you can see, that is the second layer made up of connective tissue known as the lamina propria, and the third layer of the serosa is the muscularis mucosa. Now next to serosal layer you will find the muscularis, the submucosal layer that contains the submucous plexus or the Meissner's plexus. Now as we go beyond that, you will find the smooth muscle layer which is made up of inner circular smooth muscle and the outer longitudinal smooth muscle, and after that you will find the last layer, that is the serosa or the serosal layer. Let us see the nerve supply to gastrointestinal tract. The innervation of GI tract can be divided into 2 types. The first is intrinsic nervous system and second is the extrinsic nervous system. Together this intrinsic and extrinsic nervous system, they form the third division of autonomic nervous system, sometimes referred as enteric nervous system, also called as the mini brain. Here basically the intrinsic nervous system is formed by the nerve plexuses which are locally present in the GI musculature, that is the Meissner's and the Meindrick plexus. They both constitute the intrinsic nervous system, and this intrinsic nervous system is under the control of autonomic nervous system. Now why it is important? This enteric nervous system is important because it controls most of the gastrointestinal functions, that is secretion and motility of the gut. And enteric nervous system is located within the wall of the GIT all along the alimentary canal. That is, the enteric nervous system can be found right from the beginning of the alimentary canal, that is esophagus, till the anus. Except, there is an exception for that, only the smooth muscles of the GIT, all the smooth muscles of the GIT are supplied by this enteric nervous system, except the esophagus, the upper part of 1/3 of the esophagus, and the external anal sphincter, because these two parts are supplied by the somatic nerves since they are made up of skeleton muscles. The intrinsic innervation to the gut or the enteric nervous system is composed of two well-organized neural plexuses. First is the myenteric plexus and the second is the mucosal plexus. The myenteric plexus is involved in controlling the digestive tract motility and the various movements, whereas the submucosal plexus, it is involved in sensing the environment of the gut and regulating the gastrointestinal blood flow. The intrinsic innervation to the gut performs various functions. So let us see the functions of myenteric plexus first. The major function of myenteric plexus is to control the motility or the movement of the gut. Second function is inhibition. Inhibition of the pyloric sphincter as well as the ileocecal valve is under the control of myenteric plexus. Now, how the myenteric plexus works? Whenever there is stimulation of the myenteric plexus, it will always cause the gut motility. So, stimulation of the myenteric plexus is via various neurotransmitters or tactile stimulation will lead to increase in the tone of the gut wall, as also the increase in the intensity of the rhythmic contractions of the gut, and there will be increase in the rate and velocity of the contraction. So ultimately all this will lead to rapid movement of the GI or the gastrointestinal wall. Now second is the functions of the Meissner's plexus. Meissner's plexus is involved with controlling the local secretions and as well as the blood flow towards the gut. Now, the Meissner's plexus is localized within the inner wall of each minute segment of the gut. So, it is having a proper control. Thus, the Meissner's plexus also receives sensory signals from the mucosal epithelium of the gut and the stretch receptors present on the gut wall, and it controls the secretion of local hormones and substances, absorption as well as contraction of the submucosal muscle layer. Now, the second type of innervation to the gut is extrinsic innervation. The extrinsic innervation is mainly by the autonomic nervous system. So, the extrinsic nerves are coming from the autonomic nervous system, and which has it's both the components, that is the sympathetic as well as the parasympathetic divisions of the autonomic system. So, the extrinsic innervation to the gut is via the sympathetic as well as the parasympathetic component of the autonomic nervous system. The parasympathetic innervation to the gut can be divided into cranial parasympathetic division and the sacral parasympathetic division. That is, the cranial parasympathetic fibers, they will originate from the medulla, basically the nucleus of the vagus, and this parasympathetic fibers, cranial fibers, will supply the esophagus, the stomach, the small intestine, the entire small intestine, and the first half of the large intestine. So this much of area is supplied by the cranial division of the parasympathetic nerves. Now second parasympathetic supply comes from the sacral division. So the sacral parasympathetic fibers, they will originate from the second, third and fourth sacral spinal nerves, and they will continue as the pelvic nerves, and then they will supply the remaining part of the gastrointestinal tract, that is the remaining half of the large intestine till the anus. Now the cranial parasympathetic fibers will make synapses or the synaptic connections within the or very near to the organ, That is, the myenteric and the Meissner's plexus are the synaptic junction, and whereas the sacral parasympathetic fibers, they will make synaptic connection near the organs, that is the hypogastric ganglion. The sympathetic innervation to the gut. In the sympathetic innervation, the sympathetic fibers will arise from the T5 to L2 segments of the spinal cord. Now, these fibers which are arising from the T5 to L2 segments, they will make synapses first at the celiac ganglia and the superior and the inferior mesenteric ganglia. After making synapses, the sympathetic postganglionic fibers will supply the entire gastrointestinal tract. Stimulation of different components of the autonomic nervous system, namely sympathetic and parasympathetic divisions, will cause different effects. So, let us see what are the effects of parasympathetic stimulation. Whenever there is stimulation of the parasympathetic component of the autonomic nervous system, there will be excitation of the gut muscles. So, stimulation of the parasympathetic kind will lead to excitation of the gut, and it will increase the gut motility and the tone of the muscle contraction. It will also cause relaxation of the sphincter so that the chyme can easily pass towards the aboral end. Now, it will also increase the gastrointestinal secretions as well as the local blood flow. Now, whereas the stimulation of the sympathetic division of the autonomic nervous system will cause vasoconstriction, contraction of the sphincters, inhibition of all kind of gut motility and secretions. Let us quickly and briefly see the general principles of gastrointestinal functions. So, the main activities of the GI tract are GI motility and GI secretion. So, the first basic or general principle of GI function is the smooth muscle function. So, as you all know, the smooth muscle, the GI tract is made up of smooth muscle, and the smooth muscle functions as a syncytium here. So, stimulation of a single smooth muscle will cause stimulation of the entire group of muscle present or that entire syncytium. The second special feature of the smooth muscle of the gut is that the resting membrane potential of the gut smooth muscle, it always keeps on fluctuating between -50 to -60 millivolts. Thus, this fluctuation of the resting membrane potential will always cause a basic electrical rhythm, or also known as the slow waves. So, these slow waves, they determine the rhythm of the most of the gastrointestinal contractions. Whenever a factor causes depolarization of the smooth muscle membrane, that will lead to the generation of spike potentials and ultimately motility of the gut. So, let us see the various factors that causes depolarization and hyperpolarization of the smooth muscle fibers. So, factors that depolarize the membrane are stretch of the muscle. This stretch is basically due to the presence of food. Second is stimulation by neurotransmitter, that is acetylcholine. Third is stimulation of the parasympathetic nerve. So, parasympathetic nerve will lead to depolarization of the smooth muscle membrane and cause spike potentials that will cause motility of the gut. Fourth factor that causes depolarization of the smooth muscle membrane is stimulation by local gastrointestinal hormones. Now, what are the factors that hyperpolarize the membrane? That is the causing the opposite effect. So, the factors are effect of epinephrine or norepinephrine, that is adrenaline, or stimulation of the sympathetic nerves. So, both of these factors will cause hyperpolarization of the membrane. Thus, there will be no generation of spike potentials and there will be no activity of the muscles of the gastrointestinal tract. The third basic principle of GIT is the basic movements it has. There are broadly two types of movements present all along the gut, namely propulsive movements and mixing movements. The propulsive movements like peristalsis is responsible for forward movement of the chyme towards the anus or the aboral end, whereas the mixing movements, for example segmentation movements of the small intestine, are responsible for only mixing the food. Thus, there exists a law about this called as the law of the gut that says the peristaltic reflex causes the movement of chyme always towards the anus or the aboral end. The fourth general principle is about the secretions of the GIT. The secretions of the GIT basically provides lubrication to the chyme or the bolus, and thus it protects the mucosa from wear and tear or damage from the bolus. Second, it helps in producing various digestive enzymes which helps in the digestion of the food. That is, the secretions will contain various digestive enzymes like amylase, which digest the starch and the carbohydrates, the enzymes which digest the proteins like pepsin and trypsin, and lipase and esterases that will digest the fat component of the food. Now, there are many reflexes which are operating in the GIT. Some of the important reflexes are gastrocolic reflex, enterogastric reflex, and coloileal reflex. The gastrocolic reflex is quite interesting. That is, whenever there is a presence of food in the stomach, whenever a person eats, he has a natural urge to defecate. Now, this is due to the gastrocolic reflex. So, whenever there is food inside the stomach, it will send signals all along the GI tract towards the colon, and that will cause a reflex to evacuate the colon. So, that is why in children this reflex is very much exaggerated. So that is why you will be seeing that small kids, they have a habit of eating and suddenly going to the toilet. Now, coming to the next topic, that is physiology of salivary glands. In human beings, saliva is secreted by three pairs of major salivary glands, and there are also certain minor salivary glands, but in this lecture we will be discussing about the major salivary glands and its functions. So, the major salivary glands which are present in human body are a pair of parotid glands, which are the biggest, then a pair of submaxillary or submandibular glands, and third is a pair of sublingual gland. So here in this picture you can see all the three major salivary glands in together. The first is the parotid gland, which is the largest salivary gland in humans, along with its duct known as the Stenson's duct. The second one is the submaxillary or submandibular gland, and the third one is the sublingual gland. Now let us see one by one. Parotid glands. It is the largest salivary gland that weighs around 20 to 30 grams. It is located near the angle of the jaw. The parasympathetic nerve supply of parotid gland comes from the ninth cranial nerve or the glossopharyngeal nerve. The duct that carries the parotid gland secretions is known as the Stenson's duct, that opens inside the cheek against the upper second molar tooth. The secretions of parotid gland mostly consist of water, that is greater than 90% of water. So the consistency is serous. So the secretion of parotid gland is serous in nature. Now then comes the second gland, that is submandibular or submaxillary gland. It weighs around 8 to 10 grams and it is located medial to the mandible in the submaxillary triangle. The duct that carries the submandibular secretions is known as the Wharton's duct, that opens at the side of the frenulum of the tongue. The secretion of this gland is a mixture of serous and mucus. The serous part is more predominant here. The parasympathetic nerve supply comes from the seventh cranial nerve or the facial nerve. The third salivary gland in the series is sublingual gland. It is the smallest of the all, weighing about 2 to 3 grams. It is located just below the mucosa of the floor of the mouth. And the duct, there are number of ducts, like 5 to 15 small ducts that carries the sublingual secretions, and these are known as ducts of Rivinus. The secretion of sublingual gland is again a mixture of serous and mucus component, but here the mucus nature is more. The parasympathetic supply comes from again the seventh cranial nerve, that is the facial nerve. One important note that you should take into consideration here is that the sublingual as well as the submandibular glands, they secrete fluid that contains higher concentration of proteins, so is more viscous than watery parotid secretion. So overall, you should remember that the parotid secretion is more watery when compared to the secretions of sublingual and submaxillary glands. Composition and secretion of saliva. Normally, salivary glands, they secrete about 500 ml to 1.5 liters of saliva per day. The pH of saliva is alkaline, that is, it ranges between 6 to 7.4. The composition of saliva is mostly 99% water and only 1% of solid. So the 1% of that solid component of the saliva consists of both organic as well as inorganic substances. The organic substances which are produced in the saliva are especially the salivary enzymes, namely L-amylase or ptyalin, that is responsible for the digestion of carbohydrates, cooked starch basically, and lingual lipase, that is responsible for a little bit digestion of fats. Apart from that, the organic substances like kallikrein, lysozymes, little urea, uric acid and cholesterol plus mucin is present in the saliva. The inorganic substances like sodium, chloride, potassium and bicarbonate is also present in the saliva. Let us see the mechanism of formation of saliva. It involves two processes, namely the primary secretion of saliva and modification of saliva. Under the primary secretion of saliva, that is the formation of saliva, it is done by the acinar cells. The acinar cells produce or secrete saliva by a process known as transduction. Now, the saliva which is formed in the acinar cells is isotonic in nature. What does it mean? That is, the composition of the saliva is similar. The electrolytes which are present in this primary saliva will be same as that of the extracellular fluid. The second process is the modification of the saliva. So basically what happens is that when this saliva is produced inside the acinar cell, this is taken into the ductal cells. Okay, so inside the ductal cells, when this saliva reaches, there are certain changes that is occurring. What are these changes? First is reabsorption of sodium and chloride ions from the saliva. So these two electrolytes will be removed. Thus, automatically the concentration inside will be lower when compared to that of extracellular fluid or the plasma concentration. The second change is there will be secretion of potassium ion and potassium ion and bicarbonate ion into the saliva. So ultimately the final product, the final saliva will be a modified saliva that will become hypotonic in nature. There are different phases of salivary secretion. First phase of salivary secretion is called as cephalic phase. What does it mean? It means that the secretion that is occurring, the secretion of the saliva is occurring before the food going inside your mouth. It is due to conditioned reflex, which is initiated by the sight or the smell of the food. So cephalic phase will include, that is, salivation in response to seeing the food or smelling the food, or even sometimes hearing about food. That is known as cephalic phase. Now second comes the buccal phase. So whenever, buccal means your buccal cavity. So it means whenever you are eating, putting the food inside your mouth, the secretions of the saliva are caused by the stimulation of the buccal receptors by the food. Now this is an unconditioned reflex which is partially regulated by the appetite area of your brain. The third phase of salivary secretion is esophageal secretion or esophageal phase. What does it mean? That is, there will be production of saliva whenever the food passes through the esophagus. Again, it is due to stimulation of the receptors present in the esophagus. The fourth phase of salivary secretion is gastric phase. By the name you can tell, it is related to stomach. So whenever there is entry of food in the stomach, there will be secretion of the saliva. Now the fifth is the phase of intestine, that is intestinal phase. Again, the presence of any kind. Here the food will be, it is not a normal kind of food, but whenever this phase is activated, whenever there is some irritant food present in your upper intestine, so there will be excessive salivation. Regulation of salivary secretion. Salivary secretion is controlled entirely by the autonomic nervous system. The salivary secretion is unique because it is increased by the stimulation of both parasympathetic as well as the sympathetic divisions of the autonomic nervous system. But the activity of the former is more important. That is, whenever the sympathetic stimulation is there, there will be more and vigorous secretion of saliva when compared to the stimulation of the sympathetic system. Let us first see the parasympathetic control of the salivary secretion. Here in this diagram you can see this is the parotid gland, this is the submandibular and the sublingual gland, and here you can see this is a pictorial representation of the medulla. So the parasympathetic control is mostly coming from the medulla, and this medulla is consisting of two different types of nucleuses. One is the inferior salivary nucleus and the other is the superior salivary nucleus. So let us see the parasympathetic supply to parotid glands. Now the preganglionic parasympathetic fibers for parotid gland will arise from the inferior salivary nucleus, that is from the dorsal nucleus of the ninth cranial nerve, and these preganglionic parasympathetic fibers will run via the tympanic nerve and it will make a synapses at the otic ganglia. Now from this synapses, the postganglionic parasympathetic fibers will arise and via running along with the auriculotemporal nerve, it will reach the parotid gland and then stimulate it. Now the parasympathetic supply or the innervation to the submandibular and sublingual glands. The parasympathetic fibers for submandibular and sublingual glands will arise from the medulla, that is the superior salivary nucleus present in the medulla, especially the part is called as the dorsal nucleus of the facial nerve or the seventh cranial nerve. From there, the parasympathetic preganglionic fibers will run as nervus intermedius and they will join the seventh cranial nerve fibers and leave the cauda tympanic branch as they progresses further to join the lingual nerve, and ultimately they will synapse at the ganglia present near the glands. Now after making synapses near the glands, the postganglionic parasympathetic fibers will arise from here and they will stimulate the submandibular as well as the sublingual glands. So the effects of parasympathetic stimulation will be to cause the salivary glands to secrete large volume of watery fluid that is high in electrolytes but low in proteins. Now let us see the sympathetic control of the salivary secretion. Under sympathetic control, the preganglionic sympathetic fibers will originate from the lateral horn cells of the T1 and T2 segments of the spinal cord that will enter the paravertebral sympathetic chain via the ventral roots to synapse with the cells of the superior cervical ganglia. After making synapses here, the postganglionic sympathetic fibers will run along the carotid artery branches and will supply all the three pairs of the salivary glands. So whenever there is sympathetic stimulation of the salivary glands, that will cause vasoconstriction of the glands and a secretion of small amount of saliva that is thick, viscid in nature and rich in mucus. Now come to the final topic of this presentation, that is functions of saliva. Saliva has got a very crucial role, that is first important function of saliva is protective functions. So Saliva helps to dilute the hot and irritating food substances which we eat and thus prevent any kind of injury due to that to the buccal mucosa. Now, secondly, it helps in maintaining the oral hygiene by dissolving all the left out particles which may be stuck in the mouth or corners of the mouth or in the teeth, in between the teeth. So all this, if they stay inside the mouth, they can lead to formation of bacterial activities. So thus saliva washes away all the leftover food from the mouth and helps to protect and maintain the oral hygiene. Now, saliva also minimizes the risk of buccal infection and formation of dental caries because it contains many special substances, that is lysozymes, which are bactericidal by action. So lysozymes present in the saliva helps to kill the harmful bacteria and protects from the risk of development of dental caries and buccal infections. Also, it contains local immunoglobulin, that is immunoglobulin A, that provides local defense against the harmful bacteria and bacterias and microorganisms. Now, it also contains lactoferrin, which is bacteriostatic in nature. So all these substances helps to minimize the chances of infection occurring inside the mouth. Now, saliva also helps to dilute any kind of hydrochloric acid or bile which may sometime regurgitate into the mouth from the esophagus. So, by this, if by chance there is some amount of acid or bile present in the mouth, the saliva makes sure that it provides protection by neutralizing the acid. The second role of saliva is in mastication and deglutition or swallowing. So saliva lubricates the food and it helps in easy mastication as well as easy swallowing of the food. It also helps formation of bolus by acting as a glue. The third and most important function of saliva is its digestive functions. Now, you all know that saliva also contains certain digestive enzymes, especially ptyalin or alpha amylase. This ptyalin is present in the saliva which helps in the initial starch digestion, but it can digest only those starch which has been cooked, not all the kind of starch. And also it contains lingual lipase that helps in fat digestion, basically the digestion of triglycerides. But the action of this amylase is very limited because the duration of the duration during which these enzymes can act is cut short, because whenever you are chewing the food, these enzymes will be there in the saliva. They will help in digestion of starch and small amount of fat. But as soon as the food reaches the stomach, due to the increased acidity, these enzymes will be inactivated. So there will be partial digestion of carbohydrates and small amount of triglycerides which will occur due to the salivary enzymes. Now, the fourth function of saliva is saliva acts as a solvent. Hence, it helps to stimulate the taste bud. The fifth function is saliva helps to lubricate the oral mucosa, and thus by doing this, it helps in facilitating easy movement of the lips and tongue so that proper speech can be formed. So, under dry conditions, when there is less saliva, there is difficulty in producing proper speech. Now, the sixth function is excretory function. Like many substances like heavy metals, certain thiocyanate ions and certain drugs like morphine, alcohol are excreted into the saliva. Now, the last function of saliva, but not the least, is role in temperature regulation. During dehydration, the salivary secretion is reduced, which induces thirst. So, it helps in maintaining or regulating the temperature of the human body. Thank you.